So thank you for inviting me. I didn't know about this group, and I already had a chance to talk to some really interesting researchers here. So it's creating new opportunities for interaction. So I do a lot of things, as Amy mentioned. And one of our areas of research is TB. So we do both basic biology research related to TB. We study pathogenesis, we study the mechanism of persistence, and how these organisms come out of latency to cause disease, active disease. And when we try to use what we learn in the bench to develop new diagnostic tests. Okay, so that's what I'm going to talk about today. And so, I know this is a point of care interest group, right? And one of the points I'm going to make is TB is one of those rare infectious diseases in which the concept of point of care probably will not apply. And that's the point that I'm going to make. Okay, and so you have to think different ways, out of the box ways. So if you look at this map, this was published in about 3 years ago. It's an interesting way to depict what's going on. So it's actually TB cases not detected in 2009. And so you can see these countries with the dark purple. You can see where they are. And even in the US, about 1,000 to 10,000 cases of TB are not detected. Okay, and this just tells you the difficulty in diagnosing this. These are the different types of diagnostic tests that's evolved since the early 2000s. The diagnostic tests go back to the early 1900s. So what do you think is the first diagnostic test that was developed for TB? One of the first ones.
Just clinical signs.
Right, clinical sign, exactly. Right, and then you had microscopy, right? Robert Koch, discovered Mycobacterium, caused with tuberculosis. They developed a way to stain the Mycobacterium so you can look at it in the microscope. And that's still used today. In fact, that's the diagnostic test that WHO advocates right now in most developing countries. Okay. And then the chest X-ray got invented, right, in the 1930s, and that's still used today. Okay. And so for a period of about 100 years, there was really nothing new until the technology of nucleic acid amplification started to be applied. Okay? And so, and I just want to make a point at the outset that serologic tests, I was talking to Felix earlier, serologic tests for TB, there are all kinds of serologic tests that are commercially available from these unreliable companies, and they're totally useless. Yeah, I should just say that. And WHO specifically says that these serological tests that are designed to target proteins are not to be used. Okay, and I use the word protein because we have a serological test, but we don't target proteins. And then there's this gamma interferon release assay, and this is a new test that's designed to detect latent TB infection. But again, it's not really recommended for countries low and middle income countries. So you heard about the GenExpert, right? So this is a very sensitive test. It was developed by Cepheid, and this was endorsed by WHO in 2010. So you have, this is what TB looks like. It's primarily a disease of the lungs. And so you cough up the sputum, put it into a container like this, and this can then be put into this cassette. And this little cassette contains all the chemicals that release the DNA out of the Mycobacteria and remove all the other cells that are in the sputum. And this cassette goes right into this instrument, which is basically an RT-PCR apparatus. Okay, and so there's hardly any sort of potential for contamination. Everything's just done in this cassette, and then just throw away the cassette after you use it. Okay, and then within hour to 2 hours, you'll see the amplification of if there is a Mycobacterium there, and the instrument will tell you that you have Mycobacteria or not. And it's also designed in such a way that it can detect resistance. So what they do is they target resistance against rifampin, which is one of the primary drugs used to treat TB. Okay, and the reason they select rifampin is because a lot is known about the mechanism of resistance against rifampin. Basically, more than 90% of resistance to rifampin is due to mutations in the RPO-B gene. Okay, so you target the RPO gene. If you see mutations there, then you know that it's resistant to rifampin. And the reason they picked rifampin as a marker for multi-drug resistance is because it's been at least it's often stated that if an organism, a mycobacterial organism, is resistant to rifampin, that's a marker for multi-drug resistance. It means that it's resistant to other drugs. Okay, that's why they use that. That may not be true, as I will show you. A lot of these tests developed to detect drug-resistant TB targets rifampin, RPOB. And so, expert, there was a nice systematic review of GenExpert, 18 studies looking at 10,000 patients, and they compared the results to those that were smear positive, meaning acid-fast test I mentioned earlier. The sputum is acid-fast positive. The pool sensitivity is about 90%, and specificity is 98%. So specificity is very good. It's able to distinguish TB from all the other potential targets. If you look at the smear negative samples, the sensitivity goes down to about 75%, which is still pretty good. Okay. And the limit of detection by GenExpert is about 131 bacterial pieces of bacteria, okay, which is a lot better than smear microscopy, which requires about 10,000 CFUs per mil. Okay, but it's not as sensitive as culture, which can detect up to about 50 CFUs per mil. So why don't people then use the culture?
It takes 4 weeks.
Right, it takes 3 to 4 weeks for you to grow it. Okay, and that's why, just too late. So if somebody has HIV and TB, by the time you have the detection of the bacteria, that person will be dead. Okay? And so, you want something you can detect, right away, within the first couple of days, if you can. And GenExperts, like I said, can do this in a matter of hours. And so, pediatric TB is a little more difficult to diagnose. And the reason is because, I don't know if any of you have kids, but you can't bring up sputum in kids. They'll give you spit, okay? But they won't give you sputum. So if you can't get sputum, you can't make the diagnosis. So there are all kinds of invasive things people do to kids to get the sputum. So if you do that, sensitivity is about 65 to 75%. No, not as good as adult sputum, but it's better than nothing. And then if you have HIV infection, the sensitivity is about 84%, but the smear, in HIV infection, the smear microscopy is pretty low sensitivity. And then if you look at extrapulmonary TB, meaning TB outside of the lungs, then it goes down as low as 25%, as far as nice. So it depends on where the TB is. Okay, if it's in a more accessible place, like lymph nodes, like under the chin or under the armpit, then it would be pretty high. But if it's in a deeper organ like the liver or some other places, it would be more difficult. Okay, and so in terms of the impact of this GenExpert, median time to start a treatment for smear negative patients is about 56 days to 5 days, which is pretty good. Okay, you can really reduce the time to start a treatment. And then, and if you look at the rate of untreated patients, it's about 40% without GenExpert. It goes on to about 15% with GenExpert. So it's really making a contribution. Rifampin resistance, it only takes 1 day with GenExpert, 20 days with this another method called line probe assay, which is an immunochromatographic test where you have all these pieces of DNA embedded in a piece of paper, okay, paper matrix, and then all the possible mutations. Then you amplify the product and just apply it to this. Okay? So it's more like a Southern blot test. Okay? It's a modern version of Southern blot. But it takes about 20 days. And then 120 days with the conventional drug susceptibility test. So, but there are limitations. So insensitivity in mixed infection resistant strains. So if you have, so in any given sputum, not every strain in that sample is resistant. It's a mixture of some that are resistant, some are sensitive. And so you need about 65 to 100% of the population to have rifampin-associated mutation for it to detect it. Whereas the standard test, it's called a proportional test, you only need 1%. Because if 1% of the entire population is resistant to a particular drug, that person will not do well clinically if you give that drug. Okay? So you have to be able to detect as low as 1%. Gen expert can't do that. And it cannot be used to monitor response to treatment, or treatment failure, or relapse. Why is that? If somebody relapses from TB, right, after treatment is completed, and you do the gen expert, you'll still be positive, right? Even though that, because the bacteria came up, okay? And so, or if somebody is actually cured, you can't really tell for sure that the person was cured, because the DNA will still be there, even though the bacteria is dead, okay? So it's high sensitivity is actually a detriment. So you can't monitor response to treatment. So the standard way to monitor treatment is after 2 months of treatment, you take the sputum of the patient, you look at it under the microscope. If there's no bacteria, then you can say this person is responding correctly. Okay? But very often that doesn't happen. It's still, you can still see the bacteria. Okay? But if you do gen expert on those samples, they'll always be positive because it's so sensitive, right? That you'll always pick it up. And then what do? Do you continue the same drugs? Or do you stop because it's positive and switch to some other drug? So you can't make that decision. So that's the problem. And there are a lot of false positive tests with a rifampin-resistant isomized susceptibles. As I said, not all strains can be detected by just targeting rifampin resistance, because some may actually still be sensitive to other drugs. And so, and the positive predictive value, like any infectious disease, depends on the prevalence of the disease. So it's 90% if the prevalence of rifampin resistance is more than 15% in a community. It goes down to about 50% if the prevalence is low, like in the US. So if you did gen expert to diagnose TB in the US, it's going to have much lower sensitivity. Okay. So in low multi-drug resistant prevalent areas, which involves 81 countries, up to 40% of rifampin resistant TB turn out to be isomized as sensitive. So you're going to miss a lot. And then in terms of the cost, that's an issue too, right? So, CEPHED went into an agreement with this organization called FIND, Foundation for Infectious something. It's a WHO foundation. I can't remember what it stood for. But anyways, so they negotiated the price, and if the estimated annual global usage of these cartridges exceed 3.7 million, then you can sell it for a price of $10.72 per cartridge. Okay? And then in just a couple of years ago, PEPFAR, the Gates Foundation, negotiated and said that to reduce it to $9.98. That's still not that cheap, by most developing country standards. That's $10 per cassette. Okay? And sometimes you have to do 2 per patient, because one, you want to detect whether it's TB or not, and the other to detect whether it's rifampin resistant or not. So that's $20. Okay, that's not that cheap. So that's one of the limitations of this very nice diagnostic test. So can this be sustained? So in South Africa, if you scale up Gen Expert, it will increase the cost of TB diagnostic program by 53 to 57%, just using this issue. In India, if you provide this test, just 15% of suspected cases, it will exhaust the entire TB control budget in India. So it's only going to be used for 15%. Okay? And then will the increased cost of implementing Gen Expert be offset by the overall reduction in cost of TB management? That's the argument that they would make, right? But it's not clear. Will it facilitate successful treatment in South Africa? It will require 4,020 instruments if the test were to be used at point-of-care facilities at the cost of $107 million a year. And there's also the problem of completion of treatment, default, and relapse that I mentioned earlier. So, can this test really reduce the TB burden? Okay, so that's the question. And this is a test that has to be used at point of care, right? You can't just take this home in somebody's house. So the question is, so now I'm asking a general question, okay? How much of the global TB burden is attributed to failure to diagnose TB at a point of care? So will any point of care diagnostic test contribute to the reduction overall reduction in TB. This is a situation where you have to deal with. This is where TB happens. This is Nairobi, which is the largest slum in Asia. Okay, in Bombay, located in Bombay. And then you have to take the test to this kind of setting. Yeah, are we ever going to? Do you think these people, when they develop TB, get to a point of care? They have a lot of other things to worry about. Okay. And on the average, in places like this, it takes about 6 months to a year after the onset of symptoms of TB for these people to even get to a health post. And even when they get to a health post in India, it takes on the average 3 visits to these health posts before the diagnosis of TB is made. Okay, so let's say you put this, they're very inexpensive, rapid point of care diagnostic test in some health posts. Is that going to solve the problem? So what we need is a test where we can just take it to the community like this, go directly in there, test it. Okay, but if you do that, you're not going to diagnose a lot of TB cases. You're not going to see a lot of people having undiagnosed active disease, even if you go in there. Okay? So what's the solution? And that's what you have to think about if you're going to develop a diagnostic test. That's the challenge. So this is what happens. So when somebody develops TB, okay, as I said, on the average, it takes about 6 months to 1 year for this person to have the diagnosis made. During that period, this person will infect, on the average, 10 close contacts. Okay? And so you can treat that person, but this person infected 10 other close contacts. And then that person, from this group, from these 10, 1 in 10 will develop disease again. Okay? And then that person will go on to infect 10 close contacts. And from those 10 infected people, 1 will develop TB, and that person will go on to infect 10. So you have this vicious cycle here, right? And entire world's program, TB control program, is directed at treating this guy. And if you do that, you never catch up, right? You're just going around in circles. And that's the problem. And all the diagnostic texts right now are designed to target this guy, and not this guy. What we need is to target this group and be able to predict who among those people will go on to develop the active disease. Then you treat that person. Then you can avoid this. You can not only avoid this guy from getting TB, but all of the contacts from getting TB. So you have this chain reaction. Okay? And this is what the United States does, and this is one of the few things the U.S. does correctly. Okay? They treat latently infected people in addition to the people with active disease. And this is not done in the rest of the world. Okay? So, many of you probably have skin tests, positive skin tests, right? How many of you ever got treated for isoniazid? No? Nobody got treated? I got treated for 6 months. Because when I was a teenager, I grew up in Japan, and I got BCG. Because that will also give you skin tests, but my father didn't know that. So when the doctor tested me and I was positive, I ended up getting treated for 9 months actually. Okay? I probably didn't need. But then subsequently when I started medical school, I was tested again and I was still positive. And I shouldn't be positive because the effect of the BCG wanes after many years, but I was still positive. So then I was actually very proud that I got infected somewhere in the world. I lived in many different parts of the world. And I held that as a like a badge of honor. Battle scar. And then 3 years ago, I had myself tested with a new test called quantiferon, okay, which is specific for TB. And it doesn't react with somebody who had BCG. And I was negative. And I was devastated. I wasn't infected after all. So I used, in my class, I always tell the students about this. For many years, I was actually telling a lie. And I found out. So, but it turns out in Japan, in retrospect, the reason I think mine was still positive is they give it multiple times. Most countries, they give it only at birth. Okay? But in Japan and many other Asian countries, they give it before you start school, and when you're a teenager again. So the entire program is to maintain skin test positivity. It's the exact opposite of the US. Okay. So, so this is the sort of reality. There were a series of studies where they looked at delayed treatment. So time between the onset of cough and first healthcare provider contact. It took anywhere from 7 to 69 days. Time to correct diagnosis, even after healthcare contact, was 2 to 120 days. Okay? And onset of cough to initiation of treatment was a mean of 21 days in one study in Pakistan, 136 days in Tanzania. Another study, same thing. In the U.S., it was 35 to 89 days in 2 studies in the U.S. So it took longer in the U.S. than in Pakistan after the onset of cough to initiation of treatment. With all the technology we have to diagnose TB, it still takes almost up to 3 months before the diagnosis is made. Okay? Now, another study. This was another study done in 52 studies. So patient delay, onset of cough to healthcare provider, 5 to 162 days. Healthcare provider delay, 2 days to 87 days. In low-income countries, it was about a month. High-income countries, not that much difference. Okay? You can see technology really doesn't make any difference because the technology is not applied in the proper place. So we need to consider these: failure to diagnose and TB at point of care, delay in seeking health care, failure to consider TB in the differential diagnosis point of care. If somebody comes into a clinic with lung disease, 90% of the time it's not TB. Okay? There are all kinds of other things that cause lung disease. So the important thing is, of all those lung diseases, which one is TB? And that's called differential diagnosis. Okay. And then treatment failure, either failure to treat completed treatment or relapse. Can these problems be addressed by improving diagnostics? And I think you can. So what would make a difference? Biomarkers. There's a totally different way of going after infectious disease. And you can use biomarkers to diagnose active disease, and that doesn't rely on directing the bacterial components. And you can use this to monitor response to treatment, and also to detect delayed response to treatment. You can also maybe be able to predict who among the latently infected people will progress to disease. 1/3 of the world's population is infected with this organism right now. Right now, we don't know who, when, where these infected people will go on to develop active disease. But that's why we can't give drugs to 1/3 of the population of the world. But if you can identify people who are going to develop active disease, then you just give the drug to those people, right? And that's where the diagnostic test is needed. That's much more important than just diagnosing active disease at the point of care. Ability to predict relapse, pediatric TB, as I mentioned before, TB and HIV, pulmonary TB, and then also for drug trials, you need to be able to, or vaccine trials, you need to be able to do this. Ability to detect correlates of protection with that. And then drugs. So what is this? What disease is this? Larry knows. This is smallpox. Okay? This is an example of a biomarker-based diagnosis. All you have to do is just look at the face of someone with smallpox and say that person has smallpox. That's a biomarker. Okay? And that enables people to then do vaccination around people with this kind of clinical manifestation. Okay? So if we can have a test like this for TB, then it would be useful. Just look at the person with TB and say that person has TB. Of course, we can't do that for TB, but we could identify a biomarker. Okay? Just take the blood or urine or something of a person and say, this guy has TB, or is going to progress to active disease. So you can use this to monitor response to treatment, predict those at risk for progression to disease, predict those at risk for relapse, and then treat those risk groups. Okay? And this requires not point of care, but point of residence diagnosis. Oops. So my lab studies the Mycobacterium cell wall. So this is what TB looks like, and one of the characteristic features of this organism is that 40 to 50% of the cell wall is composed of all kinds of lipids. This is what makes it so difficult to treat, because it's impenetrable to a lot of infective molecules and antibiotics. Okay? And one of the major components of the cell wall is so-called mycolic acids. Okay, these are fatty acids, long-chain fatty acids. And So we're learning a lot about how proteins and other infected molecules are transported across, back and forth across the cell wall. And that's our basic biology research. And we've been studying this one particular set of genes called MC operon. It's a cluster of 13 genes that we now know encodes the recycling of mycolic acids in the cell wall. Okay? And we knocked this out. We knocked out this gene here called MC1A, which then knocks out the entire operon. Then when you put that mutant into a mouse, the mouse is unable to make tight granulomas. This is the wild type. You see this nice discrete granulomas, okay? But here you can't really see the nice tight granulomas. And if you see something like this is going to induce some sort of host response, cytokine response, that's distinct from this. Okay? And that mutant that we created, actually, the wild type undergoes that stage during the course of infection, naturally. Okay? When you infect the mouse, for the first 8 weeks of or 4 weeks of infection, that operon gets turned off. Okay? Which is just like the mutant that we created. During that time, we predict that there's some sort of host response, okay, to this kind of change in the cell wall. We also knocked out this gene called MC1R, which is the transcriptional negative regulator of all the genes here. So if you knock this out, all these genes get constitutively turned on. Okay? And if you look at this, oops. If you infect the mouse, the mutant, look at what happens. It not only grows very rapidly, but it kills all the mice by 6 weeks. It's hyper-virulent. Okay? This is what typically happens with wild type. It goes up and then stays in this kind of plateau pattern for many weeks. Okay? But with this mutant, it just kills the mouse right away. So again, if you do this, you're going to see some sort of host response, right? That you can maybe measure. And this is what happens in 6 weeks. You see this lung? The entire lung is just filled with granule. So the mouse dies because it can't breathe. There's no airspace. Okay? And this is the exact opposite of what happens with other mutants, where they can't form the granulomas. So the bacteria keeps multiplying, and it kills the mouse because of the overwhelming infection. Okay, so you have this whole spectrum. And each of the spectrum will induce certain type of immune response that we can measure. Okay? This also happens naturally. Okay, this gene gets turned on and off during the course of natural infection. Okay? And so one of the most interesting discoveries we made in the last year, turns out Mycobacterium makes biofilm. Okay, I didn't know this. It turns out then I started looking at the old literature, and sure enough, there are a lot of old literature, I guess I didn't see them, that show that Mycobacterium makes biofilm. But what we didn't know, what was new, was that operon mutant that I talked about, the mutant operon, okay? We studied the lipid profiles, using mass spec and all kinds of things. And one interesting observation we made was that we made free mycolic acids on the surface. Mycolic acids in wild type, okay, are usually, or always, covalently linked to these polysaccharide molecules, trehalose and arabinogalactan, okay? But in this mutant that we made, these mycolic acids were not attached to anything. They were just loosely on the surface of the bacteria. Tons of them. There were 10-fold more free mycolates in the mutant than the wild type. And then another group in Pittsburgh, about a year and a half ago, found that the biofilm was made of mycolic acid, free mycolic acids. So we went back to our mutant and grew it under a condition that would induce the biofilm. And sure enough, what we were calling free mycolic acids were indeed biofilm. So this is a mutant, okay? And day 0, they're all the same. And as early as day 12, the mutant starts making this biofilm. You see this? Lacy material? And by 23, there's just tons of biofilm. Okay? And so this mutant state that the mutant represents actually occurs in the wild type in the course of infection. So we were predicting that the mutant or the wild type during the course of infection, first 4 weeks of infection, are making this biofilm or free mycolic acids. And in another study, one of my other PhD students showed that mycolic acids actually through TLR2 mechanism can downregulate the inflammatory response. Okay? And that's why this bacteria is able to survive. It downregulates the macrophage function during the first initial phase of infection, so macrophage can't clear it. Okay? And then after about 4 weeks, then the operon genes get turned on, The free microlytes that are on the surface get re-imported as an energy source. Okay? And that's why it's able to survive. It keeps recycling. So this is one of the mechanisms for persistence. We can take advantage of this knowledge to develop biomarkers. And that's what we do. So we did a TLC, which is a way to analyze lipids of the TB cell wall. What we did was we infected the mouse with TB, okay? We treated the mouse, and then and took the blood from the mouse, okay? And then used the blood to see if there are antibodies against the lipids that are extracted from TB. Okay, you can see here's untreated mouse and treated antibodies from the treated mouse. You can see there's some decrease in some of the lipid in the treated mouse. Okay. Then we took some serum samples from WHO serum bank, and we looked at the people who are smear positive TB and then non-TB patients. These are people with lung disease other than TB, and we found that there was a small lipid molecule here that was found in all of the TB patients and in some of the non-TB patients. And we did mass spec analysis of that product, and we found this peak here. It turns out to be cardiolipin. Okay? And cardiolipin is a ubiquitous lipid that's found in a variety of cell types, including mammalian cells. The structures are slightly different. So the bovine cardiolipin looks like this, E. coli cardiolipin looks like this, and this is what the TB cardiolipin looks like. So we know that the antibody recognizes that cardiolipin, right? And so we took these serum samples from people in Uganda. This was part of a CDC clinical trial of some drugs, anti-TB drugs. And we wanted to see if the antibody response to cardiolipin or other phospholipids went down during treatment. Because if it went down, that means that that person is receiving the right drug, right? And that's what we did. So 40 HIV negative patients. So we got serum samples before treatment and after 8 weeks of treatment. And we looked for IgM antibody response against these. The reason we looked at IgM is because it turns out IgM is actually part of the innate immune response mechanism, okay? IgM is needed. You know, you have all these blood cells, right? Red blood cells that are circulating. And you know, those blood cells are constantly turning over. Where do they go? And when they die, they release all kinds of lipids, right? So these IgM is necessary to clear those red blood cells when they die. So IgM binds to the dead red blood cells, okay? And then when the IgM is bound to them, then macrophages come and eat away the red blood cells. So it's part of the innate, you know, it's part of your homeostatic system. Okay? So if any lipids go up for some reason, then these IgMs will also go up. Okay? And it has no memory. So IgM, you know, it's a B cell subset that make these IgMs, okay? And they have no memory. So you don't have to worry about people who are previously infected, like with proteins. If people who are previously infected, they'll have an elevated IgG for a long time, right? With this one, as soon as the antigen source goes away, the IgM will go down. And that was sort of the rationale, okay? And sure enough, when we did this, if someone has no TB without the cavitary disease, all went down, okay? But if they had a cavitary disease, they either stay flat or went up. Why do you think that's the case? Why do you think you see this? So if you have cavitary disease, right, it looks like this. You see this little hole here, right? That means there's a lot of inflammation taking place and there's a lot of tissue destruction. When you have tissue destruction, the tissue themselves release all kinds of lipids. Okay? So when you start the treatment, okay, then you also release the lipids from the Mycobacteria. So that's why it goes up, because you have more lipids released into the circulation. Okay? Whereas the lipids that only come from TB, okay, then it should go down. And that's exactly what we're seeing. So this is a good way to monitor response to treatment. We don't have to culture the organism. All you have to do is take the blood to occasion. If it goes down, you can say that that person is receiving the proper drugs. Okay, so you don't have to switch. If it's not going down, then you have to switch the drugs. Okay? It doesn't rely on any component of the organism. And so this was the, we published this, and so look at the sensitivity of these tests with these different types of phospholipids, these topics. Oops. And then we did a study in Haiti using ELISA, and this is a little busy slide, but basically we looked at sphingosine and phosphatidylcholine, and the cases are, case positive are those people who are dually infected, HIV positive and TB positive, okay? And those are the ones where we had the highest response, again, with this IgM response. And the controls were people without HIV or without TB, and they had lower response. And this is control positive was HIV positive and TB negative. Okay, so they had the lowest. They had no TB antigens. Okay, and so with some of the cardiolipids, we think that this is going to be useful in people who are co-infected with HIV, for the same reason that it's useful for people without cavitary disease. Because when you have HIV infection, one of the reasons why people develop TB is because they can't mount an immune response, right? So you have very low inflammation. So they don't release the lipids from the lung cells. Okay? So any lipid that you see is coming from TB, and I think that's what we're seeing. It doesn't work against all of the lipids that we looked at, like Uganda. Now, ELISA is not, really something that we can take into the community, right? And so we started trying to play with different platforms. And this one we used the standard PVDF membrane using chemiluminescent method. And we're just testing the concentration of the lipids that we need to show protection. And you can see that with as low as 0.5 nanograms of cardiolipid, we can actually detect an antibody in somebody with TB. Okay? With phosphatidylcholine, we need about 5 nanograms. For some reason, with a higher concentration of cardiolipid, we lost the sensitivity. But even this, this is chemiluminescent. This is still not amenable to point of residence type of test. So we started working with Jun Q in chemistry department. Jun Q Kim, who left to become a doctor member in Texas. And he developed this method using this transparent plastic membrane. It's called polyethylene terephthalate. It's very cheap plastic membrane. And we put the lipid on the membrane like this. This is cardiolipin. Then coming with IgM, and then we take colloidal gold that's conjugated to IgM, and then detects the outcome. Okay. Now this was not a very good result, but there's some difference. We're having a technical problem. This is where we need your help, engineers, some of you engineers, how to sort of bind the lipid to this kind of matrix surface without. So one of the problems we have is when you go through all the steps of washing, the lipid comes off. Okay, and that's why we don't see it. Whereas with the chemiluminescent, we have a much better result because the lipid binds to PVDF membrane much better. The reason we wanted to use this transparent PET film is because all you have to do is just look up like this and you'll see the spot. Okay, you don't need microscopy, you don't need anything. Just go and look at, against the light and you see a spot. Okay, it's just all, relatively cheap devices, reagents that you can use to do this. So that's where we are, and we did this, and so we call this ELISA, okay? We can also use this with magnetic beads, but you don't have to use colloidal gold. And so we did a test in Haiti, where we detected just the colorimetric, response in the membrane, and so the lower numbers actually indicate darker colors. Okay, so it's kind of opposite. It's not. So we looked at the HIV-infected people with TB, with a significant difference compared to those with HIV negative TB, and then compared to the healthy controls, HIV negative. But the problem was people with TB and healthy, without TB, healthy controls, it's not much different. Okay, so it's not very good for showing the difference between TB and healthy people. But we do see a very high response in the people who are co-infected with HIV. So we still have some technical issues with this. But this was very simple. The person who did this was an MPH student, who went to Haiti, totally blinded to the sample. She didn't know which was which. And then we broke the code, and this is the result that she found. So it's promising, but not quite there yet. But that's what we are. And so I just want to end by showing you what else we're doing. So we're actually doing an entire lipidomics analysis. So that mutant that we created, we compared the mutant with the wild type. And you can see we looked at there were almost 3,000 metabolites, and we found all these metabolites that are indicated in blue-green colors that increased or decreased more than twofold from the wild type, with p-value 0.05. And this actually really humbled us. The operon gene, it was about 13 genes, right? With difference between the wild type, 13 genes, we found more than 600 differences at the metabolite level. Okay, so we know very little about the biology of these organisms when we just look at the genetics. So now we study each of these lipids, and it's very complicated because it's not like you take a new gene or a protein amino acid sequence, compare against the existing database, right? You can say, well, that's just being created right now. The lipid metabolite bank. It's a script, San Diego. And it's far from complete. There's only about, I think, 5,000 molecules there. But we were nevertheless able to find what some of these were, and these are listed here. And so these are lipids that went down compared to those that went up. And this technique actually confirmed what we knew earlier. So here, the biggest change, increase that we saw was alpha-mycolic acid, which I mentioned earlier, which was detected before by a more simpler mass spec. But using this, we detected all these that all went up. So we are now getting ready to look at the host response to all of these lipids, okay, as biomarkers. If we can identify them, then those would be specific for TB, right? So this is the group that did all the work. So ELISA was done by Roger Snyder, who was an MPH student, and then he's now a PhD student. Ken James, the one who went to Haiti to do that work. And Junyu Kim was the postdoc in chemistry who collaborated with us. And then metabolomics was done in Dan Nomura's lab, who's in the toxicology. And his student, graduate student Daniel Clickmore, and then my postdoc, Adriana Gators. Okay, I'll stop here. Any questions? I don't have anything to show you. I mean, play around with it.
Very nice talk. The TB biofilm, do you think that's happening extracellular or inside the macrophages?
So TB, when someone gets infected, it almost always goes intracellular, usually macrophage. Okay. Right. It's only when you have a lot of multiplication, you might get extracellular, but 99% of the time they're intracellular.
So you think the biofilm is actually happening inside?
I think so. I think, I don't know if you can call it biofilm, but I'm pretty sure that the free microlates are being expressed inside of it. Okay? Any other questions?
So, gene expert, can it detect latent TB or
No.
can it differentiate between latent?
No, so that's a problem. Latent TB cannot be detected by any gene amplification techniques. It's just not enough organism. Okay. Anything else?
To try to activate this lipid somehow chemically and try to covalently bind it to the surface.
Oh, activate it?
Yeah, activate. Is it possible to activate?
I don't know. I mean, this is something all new for us. I'm not a chemist, and I'm just an infectious disease doctor. So if we could, if you know anyone who is a lipid chemist, figure out how to activate it so it will stick to a matrix, some sort of surface, then we could at least overcome this problem. But that's the major rate limiting step right now, to try to keep the lipid bound, tightly to the plastic surface.
But your antibody is specific for TB cardiolism. There's no cross-reactivity with.
There is some cross. That's why we saw the elevation in IgM, when we had a cavitary disease. So there's some cross reaction.
But it still correlates with TB, is that?
Right. So it depends on the clinical presentation. So people who are low inflammatory state, like HIV-infected people with TB, people with non-cavitary disease. We're going to do this with pediatric TB also. They have low inflammatory state. Then I think it will work. But if they have a lot of inflammation, then it's probably not going to work. At least for the phospholipids that we looked at. But for some of the other lipids that we found by metabolomics, it might work, but we don't know at this stage. That's why we want to test this against all these other lipids. So it's a different concept. You know, it's not the traditional protein immunology, but it's lipid immunology. Anything else? Okay, thank you.